How can you track and predict the location of different stocks in the ocean? Fisheries managers have used coded wire tags for years to understand the general migration patterns of salmon stock. Lori Whitecamp's work using coded wire tag data has demonstrated just how different individual stock migration patterns can be, even for fish from adjacent rivers. From 1979 to 2004, in coastal waters from Southern California to the Bering Sea, about 632,000 tags were recovered. Dr. Whitecamp analyzed the location of these recoveries to study where salmon stocks traveled in the ocean. Chinook salmon showed 12 distinct regional migration patterns. Chinook salmon from each freshwater region had distinct ocean distributions that differed from neighboring regions. Despite highly variable ocean conditions from year to year, the recovery patterns were surprisingly stable. Genetic stock identification allows more fine-grained analysis than coded wire tags. Project CROOS is exploring the relationship of genetic stock identification and ocean variables to find more detailed patterns of salmon distribution to directly benefit fishermen. So, if there are weak and strong stocks in the ocean, how can we help fishermen find the strong, abundant stocks and avoid the weak stocks?
Well, that's the essential million-dollar issue we're trying to learn about, really the distribution of the different stocks during the ocean migration. The ocean is a very dynamic environment, and fish likely behave in different ways at different times of the ocean life phase. When they may be deciding to go back to the rivers to spawn, because they have to go back to specific rivers, they likely order their distribution a lot more than at other times. We are basically trying to do enough research to get a better understanding of the connection between environmental conditions, time of year, and these different stocks as to how they may vary in terms of the distribution. We have some tantalizing information from some of our observations that they may have very specific run or river-specific aggregations at certain times, and they may be much more mixed at other times. That's a really tricky thing for us to quantify, and so that's basically why we estimated that we probably needed a 5 or more year collection of data, which would likely span different El Niños, La Niña environmental conditions, and likely give us information about how fish may behave under different conditions. It of course first began in Oregon. We had this tantalizing preliminary information, so that's why we encouraged California and Washington to join us. And now we do have, for the first time this year, we have very exciting data simultaneously from California all the way up north, and we're hoping that we can get a number of years like that. There are oceanographers, microchemists, geneticists, physiologists, all kinds of people are involved in that, and we're trying to synthesize across all those disciplines. So we would like to see if there's connections between particular environmental oceanographic conditions and where specific runs or genotypes might be.
To us, the ocean may look like just a flat, uniform place, but in fact, it's highly variable at a fairly fine scale. So all sorts of things are going on in the ocean that the salmon see in the same way you see different stores as you walk down the street. So ocean environment is critically important to salmon. It affects their survival, it affects their growth, it affects their distribution. The thing I think that excited me the most about Project CROOS was the ability to use these data to learn more about salmon ecology in the ocean. By engaging the fishermen directly to sample the fish at sea, we could have them tell us exactly where and when each fish was caught using GPS. At the same time, the fishermen can collect some oceanographic information using sensors that we put on their lines to measure temperature and depth. From this information, we can map where the fishermen have been on the ocean, in other words, where their effort has been, and where they've caught the fish. So we can contrast where they fish, where they catch the fish, and which stocks they catch where. So this gives us a much finer scale picture of stock distributions in both space and time. What we hope to find is that stocks aggregate by stock of origin, so that, for example, Klamath River Fall Chinook and Sacramento River Fall Chinook travel in slightly different areas of the ocean, and that we can discern where these are and direct the fishing fleet to those areas in real time to increase our ability to harvest a strong stock and avoid the weak stock. The seafloor has terrain just like the land has terrain, and we have topographic maps of the seafloor. We also have maps of the habitat on the seafloor. The fish really tend to congregate around areas that have relief, that have hills and rocks. Satellites give us broad-scale pictures of things like sea surface temperature, which tells us the strength of upwelling and the patterns of upwelling.
Upwellings from cool, deep water bring food, the central interest of salmon. You will note on the left that with cooler blue water further out into the ocean, the salmon, purple dots, are out there also. However, on the right, when the upwelling decreases, ocean water becomes warmer, and the cooler water is near the shore, and so are the salmon. Satellites also give us pictures of chlorophyll.
Chlorophyll is the green stuff in plankton that tells us where the ocean is growing the fastest. It's a reflection of nutrient distributions and the base of the food chain. We now also have gliders. Gliders are torpedo-shaped sensor packages that fly through the ocean from top to bottom and back, and every time they come to the surface they stop and they radio home and are given new instructions about where to go and send their data out. So from these gliders we know temperature profiles, where the thermocline is, in other words where the warm water on the surface gets cold as you go down, which is very important to the fish. The depth of that thermocline tells us a lot about the ocean habitat. We know salinity, we know oxygen levels, and that way we can identify hypoxic zones which have been growing. These low dead zones are near the bottom along the coast have been a problem in recent years, and we think that that also relates to salmon. We can also tell from the tracks that the gliders take what the currents are doing. Another way we can tell what currents are doing is from a set of models that are being developed for the Oregon coast that are very fine scale. They're called ocean process models. Ocean process models attempt to model and predict currents throughout the whole water column, and temperatures and salinities, all these factors. Potentially they fill in the gaps from the measurements that we take and give us really a three-dimensional full-scale picture of what the ocean is like. In addition to being of great scientific interest, this kind of study may help us to predict the movements of salmon stocks for management.
Michael Banks is interested in further developments in genetics as they relate to salmon migration.
I guess one of the exciting things for me is a project that a PhD student I have by the name of Renee, looking at how fish know where they are in the ocean, how they determine their orientation.
So one of the things I'm interested in is understanding how organisms can use magnetic fields for migration and orientation. And something that sparked that interest was probably about 15 years ago, standing at a tree, and this bird came back, and the bird was color banded. It's a project I was working on. It had been there the year before and the year before. That bird had flown all the way to Central America and all the way back and found its way to that exact same tree. How incredible is that?
And you did the same thing.
Awfully.
Were you at the tree on
I was, but I had a map and a compass. And they have a compass and a different kind of map, so that's something I'm really interested in understanding for animals. So fish are proposed to have magnetically responsive cells inside of their nose or maybe in their lateral line. They found the cells, they know that there are cells that contain iron, but they haven't yet been able to conclusively link those cells to a neural process.
Another exciting part of this project for me is that I see lots of application for research of another colleague of mine, Kathleen O'Malley, who studies clock genes here, which allow us to learn about the genetic aspects of the timing of some of these events, so when the fish may return to spawn or how they make decisions of when to finish their ocean migration, and that's their feeding migration, and return to their spawning migration. All these different events are controlled in space and time. In Renee's work, we after the space aspect, and in Kathleen's work, she's after the timing aspect.